Hi, everyone. Thanks for coming this week. I'm really pleased to introduce today's speaker, Andrew Gordus. Andrew's background is in chemistry and physics, but he made the wise decision to move from that to studying neural circuits and C. elegans. And in particular, he's done some really fascinating work on how the nervous system can produce fundamentally random behavior. So the randomness is actually the goal of that neural circuit. Although he still works on neural circuits and C. elegans, his lab also studies spiders, which is what he's going to talk about today. So welcome, Andrew.
Hi, thank you, Adam. And thank you so much for the invitation. This has been a really great speaker series with really amazing speakers. I'm very flattered to be included. And I hope this rises up to the great level that the prior speakers have set. So as Adam mentioned, this is going to be about spiders. And much of this work is inspired by Nicholas Tinbergen's work in the study of instinct. It's a really great book. I read it fairly late in my postdoc, and it really inspired a lot of this work with the spiders. And in this book, one of many examples he uses is a stickleback fish. And he described many behaviors as these fixed action patterns where a certain stimulus is experienced by an animal, and they have an innate response to that stimulus. However, he describes behavior not as just a collection of sensory motor programs, but as sensory motor programs or fixed action patterns that are nested within different motivational states. So one of his examples with stickleback fish, he says, well, they have a reproductive instinct. And within that instinct, you can nest a variety of different motivational states that can lead to a variety of different fixed action patterns. For example, this is an example of stickleback fish fighting. And so at the very sort of consummatory act level of behavior, you do have these fixed action patterns, which the animal produces in fairly predictable ways. However, whether or not they will perform these behaviors is a function of these different internal states that they can transition through, which influences their final behavior. And a real challenge with this paradigm is that it's very difficult to try to infer these internal or hidden states based upon behavior or based on observing behavior. Oftentimes you have to infer these states based upon these fixed action patterns that the animal is behaving. But I think a really great example of a certain type of behavior where these different motivational states aren't necessarily hidden, but reflexive or reflected in the behavioral output of the organism is animal architecture. So these are a variety of different examples of animal architecture in the natural world. This is an Indian weaver bird, a bowerbird, a bower. This is a mating structure made by a pufferfish. In all these examples, the animal goes through different stages of building these structures. So these structures are an emergent property of underlying hierarchy of behavioral transitions. And these different transitions through behaviors, these are emergent properties of underlying transitions between different neuronal activities. And I'm really fascinated by this type of behavior because this behavior operates on the timescale of hours. So these are innate behaviors, but they're not necessarily reflexive. They're the results of a massive decision tree where the animal is constantly doing error assessment and deciding whether to progress onto the next stage of building. But a real challenge in trying to understand how this could be neuronally encoded is that these structures are oftentimes complex three-dimensional structures, which are difficult to quantify. They often involve acquiring mass from the environment, and they exist in environments that are not really amenable to laboratory conditions. I mean, this is at the bottom of the ocean. And they involve organisms which are also difficult to use in the lab. But I think a really great example of a particular behavior that is amenable to laboratory observation and manipulation is the orb web created by orb-weaving spiders. I think it has a variety of different characteristics which make it an ideal system for trying to understand this type of behavior. So one, the web architecture itself reflects behavioral structure. And a great thing about this is that it's a simple two-dimensional structure. So unlike those previous structures I showed, This is two-dimensional, and it's fairly easy to quantify. It's a fairly regular structure. And the majority of the behaviors that the animal performs occur along this two-dimensional plane. And so it means you can observe and capture the majority of the behavior just by observing a single imaging plane. It's innate, so you don't have to train the animals to perform this behavior. As I mentioned before, it's constantly performing error correction during construction, so it's not like it's just reflexively going through a sequence of sensory motor programs. It's built blindly, which I was really surprised when I first learned this, that these spiders are primarily nocturnal, and they don't use their vision to build the web. There's been a variety of different work proving this. I mean, I think most convincingly is just removing the eyes of the spider or blocking them. The spider still performs behavior. The eyes are also located in the wrong part of its body. Their eyes are primarily dorsal, but most of the behavioral activities is ventral. So this is primarily built using tactile senses. And it's performed multiple times in its lifetime. So you don't really have to motivate the animal to perform this behavior. They want to capture live prey. So if you take down the web, they'll build another web because they want to eat. And just as a quick primer on spiders, so this type of web is sort of the classic web found in nursery rhymes and stuff, but it's only created by a minority of spiders. So orb webs are produced by this clade here, the Aranidae and the Elaboridae. There's a major debate about whether this behavior evolved once or twice. I'm kind of agnostic to that debate, but orb weaving is only observed in basically the Aranidae or the Elaboridae clade. All other spiders produce all other sorts of webs or no web, but this is specific to two specific clades of spiders. All right, so as an example, this is a movie we took of a spider building a web. This is sped up quite quickly. It normally takes about a couple hours to build the web. Now, right now, what the spider is doing, it's building something called the proto-web. It's called the proto-web because this structure is highly dynamic. So all the threads you see here will not be part of the final structure. It's constantly putting down a line and it's constantly putting up line and taking down line during this stage. And it's assumed that what the spider is doing during the stage is that it's assessing the integrity of its environment to assess whether this is a good place to build and trying to find good anchor points to build the web. This is a highly dynamic and not very regular behavior. And then I'll just skip to, once it decides to build a web, exits out of this stage and it starts to take down the proto-web and put down the radii, as well as the frame that will be part of the permanent structure. Builds the radii, assesses it, then it builds an auxiliary spiral. This spiral is temporary. Then it builds the capture spiral, which insects stick to. And while it puts down this capture spiral, it takes down the auxiliary spiral. So it's a fairly regular behavior. And then it goes toward the center, and then it waits. Wasn't that beautiful? They're such elegant creatures. I really enjoy watching these videos. They're very satisfying. Okay, so that's web building. All of these different orb weavers, despite the fact there are differences in preferred geometry and orientations of their webs, they all go through the same basic behavioral paradigm, where they build the proto-web, which is the temporary web. Then they build the radii, the auxiliary spiral, which is thought to be kind of a stabilizing scaffold. Then the sticky or capture spiral. During this, they also take down the auxiliary spiral. But then some species of spiders build a stabilimentum, which the species I work with it does build. Despite its name, it doesn't stabilize the web, but it's thought to be an added decoration for camouflage to occlude the sight of the spider. All right, so the fact that all these different species sort of follow the same behavioral paradigm gives weight to the idea that how this is encoded in the brain is probably fairly conserved. There's also been great perturbation work done back in the 40s, 50s, and 60s, primarily by Peter Witt, where he gave a variety of different drugs of abuse to spiders and observed that different drugs perturb the structure of the web in predictable ways. What was not done now, though, was they didn't observe the behavior. So these drugs are perturbing underlying neuromodulatory pathways in the brain, which are in turn affecting the behaviors. And so the observed changes in the web are fundamentally the result of the emergent underlying behaviors which were perturbed. Also, humans aren't the first to do this, so a variety of different wasp species have evolved to also perturb the structure of the web. So oftentimes spiders which live on the web, when they molt, they actually build what's called a molt web, which is basically a web where they stop at the radii stage and don't build a capture spiral. It's thought they do this because they're kind of vulnerable once they molt, so they don't want to capture anything that might harm them. However, these wasp larvae, they inject something into the hemolymph of the spider, which basically hijacks this internal chemical signaling mechanism, and they stop the web at the same stage. So I think this gives way to the idea that this transition between these different stages are encoded by some neurochemical mechanism. So a variety of different chemical perturbations have been done in the past, and their effects on different parts of the web have been observed. For example, caffeine, in general, causes a smaller web to be built, though it doesn't really seem to affect the spacing of the capture spiral. Whereas methamphetamine doesn't really significantly change the size of the web so much, but it does significantly change the irregularity of the capture spiral. So this, all of this really inspired me to investigate this further. So there are three basic goals we would like to address. So one, we'd like to have a statistically robust definition of behavior across multiple timescales. So fundamentally, at faster timescales, or at a greater resolution than just protoweb, radii, exotic spiral, sticky spiral, we'd like to know all the behaviors involved in those specific stages and how they're coordinated. Then we'd like to perturb this behavior using pharmacologically, physically, and genetically different mechanisms, and to monitor brain activity in a live behaving animal. So those are sort of three tall orders. And this talk will primarily address this first point. So it's a really cool behavior, I think. There's been a lot of great work done with this, but a real challenge with it is that spiders are a real pain to work with. They're cannibals, they eat each other, you have to raise them in isolation. Many of the spiders that are used are only active during a certain time of the year. They're fairly large, they're venomous, all sorts of problems. Make it difficult to use them as a laboratory species. So another sort of inspiration for me to start this was, after spending several months researching different species of spiders, I found this species, which I think is more amenable to laboratory manipulation. So one, they're quite small, so their bodies are only about 4 to 5 millimeters. They have these really long anterior legs, so the full adult is about 8 millimeters long. So they're fairly small, so we can raise a lot of them together. So we have habitats in a greenhouse on campus. We have about 800 or so spiders out in the greenhouse. We're building more habitats. We'd love to have several thousand out there. We also raise spiders in controlled conditions in the lab for doing experiments. Under these conditions, we have individual wells for them to build their webs. This particular species likes to build horizontal webs. So basically, these cylinders right in here form a substrate for them to build webs, and then we feed them a familiar model organism, a Drosophila, though we primarily use Virilis for the adult because it's a larger, meatier meal, and we use Melanogaster for the younger spiderlings. This particular species has a shorter generation time than most others orb-weaving species. So a lot of your other orb-weaving spiders you find in your garden have about a 1-year generation time, which is not really amenable to a lot of genetic manipulation. But this one has a short generation time of about 2 to 3 months. It's variable because it depends on how quickly they go through their molts. It also builds webs in arid, temperate environments. So most orb weavers you find in your garden, if you put them in your house, they're less likely to build webs because they want to build webs under humid conditions where there aren't large mammals walking around. These spiders, however, build under arid, temperate environments out in Arizona and California, so they don't mind the arid conditions of the lab, and they also don't mind the presence of humans, so they readily build in the lab. We think it's because out in the wild, they like to build their webs over pack rat nests, so I think they're accustomed to a lot of physical activity and don't mind us walking around. Another clutch feature of this particular genus of spiders is that most spiders have venom glands, yet this particular genus is one of the few where they lack venom glands, which makes their neural ganglia more accessible. And I'll get to that in a bit. All right, so these are the three things we'd like to do. I'll primarily focus on behavior. So a lot of the work is inspired by great work done by others like Gordon Berman. This is actually kind of a slide just for Gordon, where he used computational approaches to try to objectively define behavior. And by taking these approaches, he was able to define behaviors of Drosophila at a fine spatial temporal resolution and high throughput, which enabled him to quantify behaviors of many organisms in parallel. As just a quick synopsis, he took videos of flies, extracted highly informative pixels from these movies, and then quantified the temporal patterns of these different PCA modes, and basically embedded them in a lower dimensional space and picked up these different densities of highly coordinated limb movements, which basically reflected different coordinated limb movements that the fly performs. And these basically represented different behaviors like walking forward and grooming behaviors and a variety of other behaviors that the fly frequently performs when walking around in a featureless environment. And this has enabled him to do really great perturbation experiments where he can then perturb the system and observe how those perturbations change which behavioral modes these organisms access. So we would like to do the same thing with spiders. Okay, so to do that, we had to build an experimental paradigm to observe them building web. So the initial sort of assay we used involved an infrared LED. The spiders don't like to build webs under light conditions. They have to build in the dark. So we have this near-infrared LED, a USB camera, and a substrate for them to build their web. As I mentioned, they can build horizontal web, so basically this is a 10 centimeter by 10 centimeter box. And by adjusting a variety of things, we're able to generate these high contrast movies of the spider building its web under dark conditions. Now initially, actually, so this really enabled us to, just as a first pass, track the spider while it's building the web. So it's easy to see. You can easily track both the position of the spider and its relative orientation as it's building the web. So you can compare spiders and look at the changes in their position as they build the web. And as mentioned before, in our context, they also go through these different behavioral paradigms. So this first phase here is highly dynamic and not very regular. This is the proto-web stage. They spend a lot of time pausing for several minutes during the stage as they will go through and build this highly dynamic structure. However, I'm going to jump to it. After a pause of several minutes, they transition to this regular stage of web building where now it starts building the radii. You can see this as the regular motion in the radial position. Once it finishes building the radii, it spirals outward. That's the auxiliary spiral. And then it goes through and starts building the capture spiral. So it spirals inward in a very regular pattern. And so we see this frequently in lots of different spiders. So once again, it's a different spider. The proto-web stage, again, is highly variable, but once it gets to the web building stage, you see the same progression of radii, auxiliary spiral, capture spiral. There are some deviations. So for example, this spider here goes through the same behavioral progression. However, and I'll just jump to it, at a certain point, it exits out of the capture spiral right about here. It goes in, it does all this plucking of the radii, and then went right back to building the sticky spiral. We assume it sensed some sort of change in the environment or there's something it didn't like, and so it went and did an assessment and then went back to a building. This other spider down here performed this in a more extreme sense, and in the middle of sticky spiral construction, it exited out of the capture spiral and all of a sudden started assessing the radii, added a few more radii, then entered back into capture spiral production, and continued. Okay. So this is just based on quantifying the centroid and orientation of the spider. But ideally, we'd like to know what the legs are doing as well. So initially, we tried labeling the legs and tracking the labeled legs. That was inspired by work done by Ben de Beaufort and a variety of others, and sort of labeling limbs of animals and tracking those points. But the spiders really hated it. They hated having their limbs labeled, and it was a real disaster. So fortunately, a couple of years ago, these two different convolutional neural network networks came out for tracking limb movements, and so we quickly abandoned our attempts to label limbs and used these approaches. And basically, we took those movies, and, took all the frames and registered them the same orientation, and then manually labeled their legs, used that as a training set, and then use these neural networks. We used both of them, both Deep Lab Cut and Leap. They both worked fairly well. I think this example here is based on Deep Lab Cut, and yeah, they worked really great. So most of the time, it predicts the limb movements really well. Occasionally, it'll make a mistake, like right there and right there. However, they typically tend to be isolated frames, and so after the fact, we remove those frames and fill them in by assuming where the limbs are based on interpolation of neighboring frames. All right. So once we had all those limb points labeled, we then took the same approach used by Gordon Berman. We applied a wavelet transform over all these different coordinates to pick up the different frequencies of movements of these coordinates in time. So there's an example here where the y-axis is time, x-axis is time, y-axis is different frequencies. This is an example of one particular coordinate, and basically the bright pixels represent high amplitude sampling of a particular frequency. So this is a slow movement, this is a fast movement. So we can do this for all these coordinates in time, and you can see as the spider is moving along the web, the labels dropped out here, but basically these are different coordinates. It's sampling all these different frequencies as it's building the web. And so for each frame, we can take the same approach of using t-SNE to embed all these different sample frequencies and identify clusters of highly frequently sampled coordinated limb movements. So here's an example. This is an example of in the spider during the proto-web stage. It samples a variety of different densities in these lobes here. But then once it starts entering the spiral stage, it starts accessing these frequently sampled densities over here on the left. So initially, we were pretty happy about this. It seemed to work pretty well. So the goal was that we would have this action pattern density map of different fixed action patterns, then build a transition map between these and try to define behavior based upon these different sample densities and the transitions between them. However, a problem we encountered was that this approach was highly sensitive to idiosyncrasies between spiders. So right here is the commonly embedded map for all the behaviors, and it's colored according to the maximum proportion of density contributed by a single file. So predictably, the noise on the perimeter, these points are from single files, but a lot of the densities were coming from single webs, which meant even though we're embedding these in the same space, different webs were embedding differently in the same common space. Yes, the sparse density is represented by single webs. So we had a lot of trouble trying to sort of identify behaviors which are common between the webs. I'll just skip over this. And so we should really struggle to try to figure out why this is happening. And I think there are two primary reasons. So one, this approach is sensitive to idiosyncrasies from different spiders, especially since these spiders are not walking around a featureless environment. They're walking around an environment that's constrained by the structure of the web. And so as you create different structures, the behaviors are getting pigeonholed into these different frequently sampled behaviors which are operating at slightly different frequencies. The ideal case is that you have a behavior which, like this posterior limb behavior, when it's combing the web out during the capture spiral, where it has this nice regular frequency. However, many of the behaviors, the limb movements aren't as regular as this. So unlike a lot of the fly behaviors, which are sampling a lot of sort of frequently sort of repetitive behaviors, a lot of these spider behaviors, as it's maneuvering around the web, are behaviors which are not necessarily super regular. For single recording embedding, some clusters are quite interpretable. So a lot of these densities are coming from the capture spiral construction, and that's because when it's building the capture spiral, the posterior legs are operating on a very regular basis. They're kind of shaking and combing the lines out. And so in some instances, for single recording, we are able to look at these densities and identify clusters which are interpretable. However, they're hard to find across the recordings. So if you look at where on the web a certain cluster occurs, in some cases, for example, this cluster here, it seems to regularly occur at anchor points where the capture spiral is anchoring to the radii. However, many of the clusters, in fact, most of them, with this approach did not regularly occur. So for example, this cluster here is primarily dominated by this web and was just kind of randomly being accessed in these other webs. And so this heterogeneous space lets it difficult to interpret transition probabilities. So we have all these different action patterns that are being embedded in the same space, but they're embedding in different parts of the space, which predictably leads to different transition maps, which lead to these different webs. Okay, so I'll just skip over that. All right, so the intermediate conclusion was for this particular type of behavior, this wavelet t-SNE approach was able to pick up some structure, but the variability between recordings and high dimensional nonlinear nature of the transform made it really difficult to interpret the results. So we really struggled with this for a while. Initially, we thought the tracking was different between recordings, or there's some variability in the experimental paradigm. So we thought about this for a while, and we decided to take a slightly different approach and see how it worked. So with this different approach, instead of applying a wavelet, we directly aligned time series. So an advantage of the wavelet approach is that you're not limited to a single time scale. With this approach, we are limiting it to a certain time scale. So for every frame in the movie, we center a 1 second window around that frame. And then we compare that window to other frames. And then we define the distance or the similarity between them just based on a Euclidean alignment error between those different frames. It's sort of Euclidean, it's not exactly Euclidean. And the reason why I say that is because we align them, but we allow for a little bit of wiggling between these limb movements so that these different behaviors, the limbs don't have to be perfectly coordinated in phase to be considered the same movement. So we allow for a little bit of wiggle movement with regard to the phase of each limb. So it's sort of Euclidean, but not perfectly Euclidean. So we took this approach instead of the wavelet approach. And with this approach, we had a greater consistency of identifying densities that were common between webs. And while looking at the densities, many of them were fairly easy to interpret. So there were a variety of different capture spiral behaviors we saw, but also a variety of different non-capture spiral, like a right leg sweep, the sort of walking behavior, which is commonly observed during auxiliary spiral production, and this extrusion behavior, which seems to occur right when or we think it's occurring right when the spider lays down that capture spiral right on one of the radii. Another sort of nice thing about this approach was that when we look at how the different webs embed in this common space, that the general geometry is fairly similar. So they're embedding in a fairly similar way into this common space, and they're accessing these different densities at similar points in web construction. So this isn't perfectly aligned according to this web construction, but they're sampling similar densities during the radii stage, and then when you get to the spiral stage, they're also once again sampling similar densities. And so if we take a look at these densities, for example, this cluster here, and we look at where it's sampled across different webs, we can see that it's fairly well represented across different webs and represented in the same general spatial temporal pattern across these different webs. So that this anchoring behavior is fairly consistent. Another nice feature is that this density here is seen to be unique to the stabilimentum construction. So these webs that did not, this density was not represented in these webs, and these webs here, this web, and this web, these spiders did not build a stabilimentum. However, the spiders that did build a stabilimentum, we did see this a cluster appear in those webs. With the species, they tend to build two types of stabilimentum, either a linear one, like we see here, or some of them build a spiral stabilimenta. And this different density basically represented this spiral construction. So that's nice. That's something we didn't see in the previous approach. And yeah, so a variety of the different densities were well represented across webs, which is something we had difficulty observing with the previous approach. We've also tried using agglomerative clustering to define densities. So in the previous approach, you define behaviors by embedding into this lower dimensional space, and then using a watershed algorithm to define the clusters. So the clusters are basically defined by these highly sampled or frequently sampled states. So density is very important for defining these behaviors. Whereas with agglomerative clustering, that's sort of density independent. These different behaviors are sort of being clustered based upon their Euclidean distance. So it's independent of how frequently they were sampled. And using this approach, we also identify similar clusters yielding similar results. So this cluster here also captured this anchoring behavior. This cluster here represented the stabilimentum behavior. And this cluster here represented this walking behavior, which the spider frequently produces as it's building the web. All right, so taking this approach, we're now building these transition matrices to try to understand the rules with regard to transitioning between behaviors, and hoping we can use this to build a transitional map of web building behavior. We're in the midst of this right now. We'd like to see how consistent this matrix is between different webs, but the goal is to basically go through this entire pipeline and build this sort of objective measure of behavior, and then use this as a measure to compare webs and individuals between individuals and the same individual over time to understand how robust these different behavioral algorithms are. One disadvantage of this approach is that you may have observed that we're quantifying the limb movements, but we're not observing the web construction. So we intentionally illuminated the spider in a way where we could not see the web, because seeing the web was a confounding issue when trying to use deep lab cut or leap to track the limbs, because you have all these sort of lines in addition to the the limb movements. Also, trying to eliminate the web is really difficult. Traditionally, people spray paint the web after the fact, take a picture of it. Clearly, we don't want to spray paint the web while the spider is building the web. But ideally, we would like to have a measure of the web as the spider is building the web, because the web is both the sensory input into the behavior and it's also the behavioral output. So we re-engineered the system. We still have the ring LED at the top to illuminate the spider and the USB camera, but then we built a ring of LEDs below the web frame that illuminates the web at an oblique angle so that the light refracts through the web up into the camera. So this is basically what it looks like. These are the ring LEDs beneath the frame. So that illuminates the web. This illuminates the spider, the camera. And basically, if you just illuminate with the top LED, you can see the spider really well. But then if you illuminate with the ring LEDs, the spider's blown out, but you can see the web very well. And so we basically strobe these lights so that we interweave illumination of the web and the spider, so it's pseudo-simultaneous. This is all controlled by a little teensy. So by doing this, we can monitor spatial temporal changes in the web, and then try to build a sensory motor model of behavior by using both the observation of behavioral changes and changes in the web. So as far as quantifying the web, another challenge is that the lines aren't uniformly illuminated. So this is what it looks like. So what we've done is we've, Abel, my student, has written a maximum likelihood estimator to try to predict where the lines are based upon these frames. So we had a high school student, Maya Filipovitz, go through manually annotate these. And then basically the pipeline is using a combination of a Hoff transform to identify lines in the image, and then a maximum likelihood estimator. We go through and try to predict where the lines are based upon this illumination. This is the work in progress. But the goal is that we'll have these different motor states that we can identify that spider's producing, and these different web states. And these different web states are both the sensory input into the behavior and also the output of the behavior. And so by monitoring these two states, we can build a minimum sensory motor algorithm to try to see how a simple sensory motor model of behavior can, how well it can predict web progression. However, we also think that in addition to this, that the spider is going through different internal states of web progression, and that this is also influencing how the spider builds the web. And so we're eager to test how well this works and how well this internal states modeling would, how well this would work as well for defining web progression. I think a nice thing about this is that we could define these internal states not necessarily by some hidden state, but by knowing that the spider goes through these different behavioral transitions between radii and auxiliary spiral, and see how well dividing up web progression by these states changes the different sensory motor algorithms the spider accesses. Okay, so we'd like to define the behavior in different time scales, but this is all very passive. We'd like to also perturb the behavior to test how well these models predict, how well the spider, how well these models predict web progression. And we'd like to be able to perturb the system pharmacologically, physically, and genetically, and monitor brain activity. So we're toying around with a variety of different models to physically perturb the web, so that we can physically, bring something in here to perturb the web. There's classic work done by Bill Eberhard out in the field, where he showed that while the spider is building the web, if you remove part of it, the spider will exit the web building algorithm and basically go in and fix the web, and then go back to web building. Sort of like what we observed spontaneously in a few web builds. And so we'd like to know what level of perturbation will the spider accommodate in doing error correction, and at what point does it sort of abandon error correction and take down the entire web and rebuild a new web. As I mentioned, we'd like to do genetic perturbations. This sort of this part of the talk is real injustice to Jeremiah, but ideally we'd like to genetically perturb the spider. That requires knowing what the genes are. So my student Jeremiah Miller has basically been trying to assemble the genome. So far we know it has 8 autosomes and 2 sex chromosomes, and they appear to be telocentric. Sex determination, this is very common from a sex determination in spiders. So both males and females are diploid for autosomes, but the female is diploid for both X chromosomes, and the male is haploid for both X chromosomes. So males are XXOO, and females are XXXX. This single slide basically covers years of work by Jeremiah, but basically we used a combination of Illumina and nanopore sequencing in combination with this genome assembly work structure called Mazurka to assemble contigs. As a first pass, Jeremiah's contig assemblies were very good. So his N50 for the contig sizes is much larger than other published genomes. And the contig number is fairly small, which is good relative to other published genomes. Ideally, that number is the same as the number of chromosomes. 48,000 is clearly more than 10. And at this point, I kind of wanted to just go ahead and publish this, but Jeremiah wanted to get a chromosome level assembly, so we decided to try out Chicago and Hi-C sequencing. And with this approach, you don't sequence pure DNA. You build a mate pair library made of DNA fragments that are in close proximity because you assemble them on using chromatin instead of naked DNA. And so the frequency at which 2 pairs are sequenced is a reflection of how close they are in physical space. And so by doing this, Jeremiah was able to produce 10 megascaffolds of proximity, and we think that these 10 scaffolds basically represent the 10 chromosomes of the spider. So I think we do have a chromosome level assembly of the spider. And right now, Jeremiah is in the midst of finishing the annotations and doing some comparative genomics work. We also have a transcriptome. And so the goal is that with all this information, we can now leverage this information, and as a short-term goal, perform RNAi experiments by knocking down a variety of neuromodulatory pathways, which are the targets of those different pharmacological drugs that have been used in the past with spiders, to try to understand how these different pathways influence behavior. RNAi has been used in the past with spiders. And our long-term goal is to eventually introduce transgenics. RNAi has been used in the past with spiders, so we think it should be informative approach for perturbing behavior. And then in the last few minutes here, we're also interested in trying to understand how this behavior is encoded in the brain. So ideally, we have an anatomical map of the brain. This is a Drosophila brain. Ideally, we would have this. This is, of course, the result of a century's worth of work. I likely won't live that long, but we'd like to get something sort of close to this. There has been previous anatomical work with spiders. So in general, the structure of the spider brain is plesiomorphic relative to other arthropods. So as a esophageal ganglion and a subesophageal ganglion. In the supraesophageal ganglion, they have 2 primary neural pills. One is called arcuate body, which is thought to be analogous to the central complex in insects, and a mushroom body, which is thought to be analogous to the mushroom body in insects. A real challenge with a lot of spiders is that right here, PG here stands for poison gland or the venom gland, and you can see it lies dorsal to the brain here. And so if you were to cut the cuticle, that would be a huge nightmare because you don't want a popped venom gland on top of your brain because it's filled with neurotoxins. We spent a lot of time just trying to get immunostaining working. We eventually did have it working, and so this is a couple examples of cardioactive peptide green on the left here, or a latostatin in yellow. And the staining basically captures this arcuate body, banded structure, and then the mushroom body. In addition to immunostaining, we've did a micro CT scan, and are right now in the process of identifying different neural pills based on density. As a first pass, we were able to capture the mushroom body, arcuate body. Obviously, a very happy result. So in addition to this, we'd like to get fish working, which is an ongoing project. And we'd like to build a functional map of the brain as well using live calcium imaging. And that's a pretty challenging goal. There has been electrophys work done in spiders in the past. Most of it has been done on the peripheral nervous system. And that's primarily because spiders, their hemolymph is under a lot of positive pressure. So they don't have extensor muscles. So to extend their limbs, they use positive pressure and relaxation of their flexor muscles to extend their limbs. So like C. elegans, if you cut their cuticle, the hemolymph blows out. So if you cut the cuticle up here, especially with salticids, jumping spiders, where most work has been done in spiders, the brain literally blows out. And I've tried this, and it's true. It's pretty catastrophic. One group, Ron Hoy's group in Cornell, has successfully done extracellular recordings of the brain where they very carefully puncture the cuticle with a titanium wire and able to pick up these extracellular recordings. But a challenge with this work is that you have a lower anatomical resolution. So ideally, we like to do calcium imaging. So despite all these challenges, we thought we'd try to give it a shot. So this is basically we modified the protocol used for Drosophila. So we modified the dish that Drosophila are glued to. We made one custom for the spider. Ideally, we glue the prosome of the spider to this dish under saline solution, then bring the subjective in and try to monitor brain activity. Of course, everything had to be custom made. Abel very heroically 3D printed a variety of these things and custom built this Peltier setup. Another challenge with the spiders is that we have a lot of trouble knocking them out of CO2 without killing them because the effective dose and lethal dose are like really close. Also, cold anesthetizing them is difficult. They're very robust to the cold, so we use this Peltier. I think Abel brings it down to like just close to freezing, and it's sufficient to finally mobilize them. So I'm going to skip most of these steps, but basically we orient them on this Peltier to immobilize them, orient them relative to this dish, glue them to the holder like you would Drosophila, and dissect it under saline. And so here is dissecting it, and then at the end result there, you can see that's basically the brain of the spider. Now you may have noticed there are two things here. So one, since this is a yield board, the venom glands, which would normally be here and in the way, aren't here, so that's great. And then two, you may have noticed the brain didn't shoot out like I mentioned before. One other nice feature about this particular species of spider is that it's actually really horrible at walking on horizontal surfaces. I should really take a video of this, but unlike salticids, jumping spiders are very quick and good at walking. These spiders have a lot of trouble supporting their weight. So when you put them on a horizontal surface, they really drag their body because their hemolymph is under lower pressure, so they have difficulty extending their legs and supporting their weight. They prefer to be hanging on the web, and they use gravity to extend their legs. So there is positive pressure, and eventually they lose the ability to move their legs. But we can keep them like this for up to 12 hours, and they're alive, and we have access to their brain. So what we would like to do is use chemical calcium sensors generously provided by Luke Leif's group at Chinelia, and inject these into the brain to monitor calcium activity in a awake behaving spider. This project got derailed recently because of COVID. We'd like to come back to this and test how well these sensors work under this dissection prep. And ideally, we would like to have the spider perform a sensory motor, some sort of sensory motor program while we're imaging them. I think trying to have them build a web is a tall order. Ideally, we'd like to have them sort of walk on a ball. However, this organism does not like walking on a surface. So when Abel was a rotation student, we thought of an assay where we can try to keep the spider fixed and for an imaging assay, but then have it sort of fictively walk on a web. And so what he built is basically an air table, and we had the spider build the web on a foam frame. Then we remove the spider from the frame. We place that frame on this air table. Then we fix the spider to a pin and then carefully delivered it to the web. And then once it engages with the web, it can basically fictively walk along the web. So this is kind of like analogous to the fly or a mouse walking on a ball, but it's a spider walking on a web. And so at some point, we would like to combine calcium imaging with this assay. So that's basically the pursuit of this part of the lab. We have three main pursuits. We're trying to define the behavior and then perturb it both genetically and physically, and then ideally try to understand how this behavior is encoded by the brain. All this work was basically performed by three individuals. Nick Wilkerson, who was initially an undergrad in the lab and then worked as a technician for a couple of years. Jeremiah Miller, the graduate student in the lab, then Alba Korver, who took over the behavioral project and started the neuronal side of this project as well. Just as a quick plug, most of my lab actually works with C. elegans, and we have a variety of different, very exciting projects that we're doing, looking at sensory motor transformations and memory and worms. So this is the entire lab. I really like to thank several people at Johns Hopkins who are much better at genomics than me, especially Winston Timp, James Taylor, who recently passed away, Michael Schatz, who have helped us a lot with the genome assembly, Bob Johnson, who's helped us a lot with immunostaining, Bill Eberhard, who basically taught me most of what I know about spiders. He's like the word-weaving god of the universe. And then Luke Lavis and Karen Pfeiffer, who have helped us a lot with trying to get calcium imaging working. And of course, funding. And thank you for listening.
Great, thank you, Andrew. I have a quick question. Can the spiders tell when they're on a web versus on a ball by like mechanical forces, or is it just like the like where they have to position their limbs, or how do they tell what kind of substrate they're on? You mean like how they know they're walking the ball? So these spiders, they don't walk on the webs. They hang from their webs, and so. Oh, I see.
I've never seen a spider walk on top of the web. It always hangs.
Okay, so yeah, everyone, add your questions, but we already have a bunch here. So Chris Rogers wants to know whether you think the spider's brain contains an internal model of the entire structure of the web, or is it making moment-to-moment decisions based on spatio-temporally locally cues?
That's a great question. I mean, my pet hypothesis is that the entire web progression is encoded in the brain somehow. I think the ability of the wasp larvae to pause web building at a specific transition gives credence to the idea that there's some sort of neuromodulatory mechanism that encodes the transition between these different stages. But I think trying to build a minimal model just based upon sensory motor transformation, so just based upon the structure of the web and the motor program, would be a good first step in just trying to see how well a minimal model works in modeling this behavior, and then trying to layer on top this internal state to see how well that extra level improves web building, and if those sort of blind internal states match the sort of physical states we see in web progression. Yeah, my pet hypothesis is that there is some sort of progression encoded in the brain, but that remains to be seen.
Shubham Chitnis wants to know whether the geometry of the substrate affects the structure of the web. Can you get like different webs from different substrates?
Yeah, so they're fairly amenable to different sizes of substrates. All your different species of spiders have a general range they want to build across. And typically on the order of a few dozen body lengths. So if the substrate is too large, the spider won't build a web. If it's too small, the spider won't build a web. It tends to be unique to different species. For example, the Darwin spider Madagascar prefers to build webs across rivers. This particular species likes to build webs that are basically on the order of about a foot large or tens of centimeters large. There has been work changing the geometry of these frames, and the spiders are fairly good at accommodating different geometries, and building the capture. If you put it in sort of in a rectangular structure, it'll build an elliptical orb to try to sort of accommodate that space.
There's a question that was on here earlier but got taken off, but I thought was interesting is, do individual spiders have, like, how much variability are the webs from individual spiders? If you had a spider build the same web again and again and again, will it look relatively similar or will it look totally different?
That's a good question. That's something we'd like to investigate further. So in some cases, certain spiders seem to be relatively consistent in how they build. However, there's a temporal progression, or temporal change in how they build webs. So initially, they're fairly robust at building, but then once the females reach sexual maturity and start laying eggs, their webs become more variable. They're called senile webs, and I'm not sure if that's because they're sort of losing the motivation to build webs, kind of like an octopus. Like after they mate, they kind of lose motivation. Males stop building these webs once they reach sexual maturity. They primarily live off the female webs. But that's something we'd like to investigate further, like how idiosyncratic are these webs, between individuals or within an individual. And I think by assembling this experimental paradigm, we can use objective measures to try to define variance between and within between different individuals.
Dinesh Nadazan wants to know whether there are other major sensory inputs to the spider, and how about vibrations to the web? Can they be used to estimate like the stability or other aspects of the web that they built so far based on the vibrational frequency, perhaps?
Yeah, that's a great question. So I know a few talks ago, there's Mahati talked about vibrations, and so yeah, they're very sensitive. They're very sensitive to web vibrations, and we have taken some high frame rate videos of them moving along the web, and that's something we would like to investigate further. Is we'd like to sort of observe the vibrational spectrum of the different lines and experimentally manipulate the web to try to see how different vibrational information influences the behavior. And it's thought, so during the proto-web construction, it probably went too fast, but there are times when the thread is loose and then the spider tightened it and anchored it again. It's also thought it uses vibrational information to identify prey in the web. When the prey hits the web, it's this massive global perturbation, but then oftentimes the spider will pluck the lines of the radii and then turn toward the prey, so it's thought that it probably plucks the lines and senses the higher vibrational line, which indicates the shorter line, and that's where the prey is. So it likely does use vibrational information, and yeah, that's something we'd like to investigate further with regard to how it influences web construction.
Molly Lou wants to know whether you think you'll be able to do an unsupervised clustering of the web building stages, for instance, using the spider position or orientation or recent silk geometry to classify as radii or capture spiral, the same way that you've done with the spider posture. So cluster the web building stages rather than the posture.
Yeah, the web building stages based on spider position is really easy to do just because of the regularity of those different periods in web building. So when it's building radii, there's this high amplitude, high frequency signal in the radial coordinate, and then when it switches to auxiliary spiral, the radius increases, and there's a high power in the rotational coordinate, and then when it does the circular spiral, you see that again. So those periods of web building are very easy to define just based on the coordinates. You don't really need to use any sort of higher order manipulation of the data to identify those stages.
I don't know, this person at U2UU asks whether the web consists of only one type of thread or there are multiple types of thread for different functions.
Oh, that's a great question, and I forgot to mention that. So Orb weavers, relative to other spiders, have a greater number of different silk glands. So they use different types of silk for different parts of the web. So the radii are built with a very strong type of silk that's not adhesive, whereas the capture spiral is a different type of silk that comes from a different gland that is sticky but not as strong. This particular species that I work with, the capture spiral is different than your typical orb weavers. So the classical orb weavers, the Anidae, their capture spiral is hydrated, so there's like a sticky glue on it, and it needs to remain hydrated to function, which is why you primarily see these spiders in humid environments. Whereas the species we work with is in an arid environment, it doesn't use a hydrated type of silk. It uses this criblet silk that's kind of combed out, and it's very fine, and it adheres to the wax on insects. It kind of like absorbs into it. So the different web stages are also represented by different types of silk that the spider uses during web construction. And that also influences how the spider performs error correction. So if it's exited out of radii construction, it oftentimes kind of uses a slightly different behavioral mode to fix it, because it's less likely to switch the glands over. It wants to keep using the same glands it was using before. So it uses a variety of different types of silk.
Can you tell, are the different types of silk stereotyped enough that you don't have to, that you know exactly when they're producing it, or can you tell based on like the reflection or refraction of the light, or the, I guess, the different movements the animal makes when it's producing silk?
It's difficult to tell just based on looking at it. However, the capture spiral, the criblet silk, is easy to define because the spider has to comb it out, and it has this kind of woolly like texture. I mean, the canonical paradigm is that it uses different types of silk for different parts of the web. However, there's some debate about that, about whether it's truly exclusively using different glands, or whether it's more subtle than that. That it might use a mixture of the glands as it's building the silk based on the conditions. There's a researcher, University of Akron, Blackledge, who has investigated this far more than I have, and it's an active area of research to figure out how consistently it uses these different types of silk in different web stages.
James McLeod asks whether the neuromodulatory changes associated with egg laying are well known, and could they be a way to uncover? Whoops, lost us. Could they be a way to uncover web production networks?
Egg laying?
Yeah, maybe James can clarify in the comments. I assume he's asking the egg laying, the neuromodulatory changes for egg laying might correspond to changes in web production or the way the web is built.
I don't know. I mean, one line, this is kind of similar, but one experiment we would like to do is to give ecdysone agonist or antagonist to the spider and see if we can pause it at that transition between the radii and the axillary spiral, because we assume that some sort of ecdysone signaling might be used to influence that transition, since when the spider molts, it also has this pause in web building.
Bitu wants to know whether you have a sense of what features your spiders are using to select the site to initiate web building, and what the algorithm is that goes into that initiation decision. To
identify a good location to build a web?
Yeah, like how do they decide, like, I want to build a web here versus somewhere else? Is it just like,
I mean, there's a lot of work with this out in the field for different species, and ideally they try to find a location where there isn't too much movement, so they can stably build the web. They're very sensitive to perturbation during the proto-web stage, which is something I didn't mention, but in the proto-web stage, if you perturb them during that stage, they're less likely to build the web, presumably because they've decided, well, this is not a good place to build. They're less susceptible to perturbation as they progress. So if you tap the frame during the last final stages of web building, they just kind of like, well, I'll just keep building this because I'm really committed to web building. I'm just going to finish this web. So it's assumed that during the proto web stage, they're assessing the environment to decide whether it's a good place to build the web. We think they're also assessing how sticky the anchor points are. So early on, when we did this, we scuffed up the plastic to provide it with a substrate for building the web. And initially, where the center of the web was highly variable under these conditions. But then when we put construction paper down, the web seemed to be a little more consistent. And so perhaps that was because they had consistent anchor points for setting the web. So it's something we'd like to do is manipulate the integrity of anchor points and see if that influences how and where they build the web.
Brendan is asking whether the spider, you said the spider was alive for up to 12 hours in your dissection. Were the legs also functional for that whole time? He's wondering in particular whether loss of limb function due to pressure differences may be an insurmountable problem for the prep. And apologies for
so there are a variety of different steps we want to go through. So one is just loading the dye and monitoring signals. And two, initially we're interested in just looking at sensory responses. So can we see, neural responses in the optic lobes of the spider and responses to touch and vibrations along the legs. It progressively loses function of its legs over about an hour. We've also toyed around with just removing the cuticle and then putting glue on top, which is an approach that's been used in Drosophila, I think by the Schnitzer group and a few other groups. So with that approach, in principle, we would inject and then put the UV cured glue over, and that would basically seal the prep up and provide us with a window into the head. So there's a variety of different steps we need to get to before we get to the ultimate experiment of having you do something. But I think at first, just observing sensory responses would be a good first step.
And then Frank asks the last question, which is, what orb weavers can patch the part of the web where the prey is captured. He asked whether you've thought what goes on in there, and I assume he's asking, are they doing different behaviors, or are they just kind of relitigating the same web construction behavior they did when they were working on that in the first time, or are they doing something new?
I'm sorry, what was the first part?
Orb weavers can patch the part of the web where prey is captured?
Right, that's right, yeah. So they'll patch up parts of the web where prey is captured, or if kind of poke a hole in the web, they'll go and patch it up.
So is something unique going on there, or is there like unique patching behavior, or is it like a similar behavior as the web building?
That's something we're really interested in. So initially, before the whole COVID nonsense, we have a high school student in the lab, and her summer project was to poke larger and larger holes in the web, and just observe how the spider patched it in. And then at a certain point, if you remove too much of the web, the spider will just take the whole thing down and rebuild it. So we're curious to know, how great of a perturbation is sufficient to cause that changeover from patching to rebuilding the whole web. And then another parallel project we wanted her to do was to have a spider start building a web and then stop it and just remove it from the web, and then put a different spider on there and ask, can the spider assess the status of the web and continue building the web, or does it have to go through the entire behavioral progression? So we'll just take it all down and then rebuild it. So maybe we'll do that next summer.
Hoping there's less COVID, I guess.
Right. Yeah.
Okay, that's it for today. Thank you so much for coming, Andrew.
Thank you.
Thank you, everyone, for being here. We're not going to be, there's not going to be a seminar next week, but in 2 weeks, Lindy McBride will be talking about mosquitoes. So please come to that. Thanks, everyone.